I appreciate everybody coming who's made it here so far. We're kicking off the LBRN annual meeting today with the first ever core core session where we present relevant technologies and services that the LBRN offers to our investigators as well as nearby campuses. So with that, I'm gonna lend the floor to Yong Lee, who is the Director of the Molecular and Cell Biology Resource Corps. And
Morning, everyone. My name is Yong Lee, and I'm leading Molecular and Cell Biology Resource Corps. We have brief time about introduction of this core, and actually because of many needs, we featured this talk with some translational study part. Okay, the mission of this core is provide best possible technological and logical support for exchange of information. This is very hard to understand what it means. If I understand, the way I understand is we all are like biomedical interest groups. We every day feel that technology involved in biomedical science is ever advanced, and it is so hard to catch up every day. And it also means it is very costly. So what this core can do is we can unite all those possible technologies, at least within states, as make one single united group and provide those technology and the service to your local researchers, not only LBRN researchers, also some other state researchers. And we can provide the service, consultation, and the training. That's the whole point of our core. So there is some, we have established network of technology source using LSUBR as a technology hub. There, of course, School of Veterinary Medicine has most facilities, and so that is pivotal role. And there is also shared instrument facility in LSU, and the Center for Computational Technology, CCT, and the Center for Advanced Microstructure and Devices, CAMD, as additional source of technologies. And then in the not only LSUBR, there are in statewide, we have 5 NIH COBRA programs and the 1 NIH super fund. That means each COBRA program and the big NIH program has its own team of research, and their own research core for team-oriented specific researches. Some of them we are overlapped, but there is some specialized research we can use, okay? Now in LSU alone, we are operating gene labs and the protein labs and then molecular immunopathology lab and the structural biology lab, statistical service unit, and the animal service welfare. So that we can use. And then in LSU shared instrument facility, we have electron microscope, cryo-EM facilities, we have NMR and the mass spec facilities and the genomics facilities. Combined together in gene lab, what we can do is DNA recombinations and of course conventional sequencing plus chip DNA prep and sequencing, epigenetic sequencing analysis, mRNA preps and qPCR analysis and the sequencing for transcriptomics, microRNA preparation sequencing. And there is also a very active supply of high-use molecular biology reagent. We have in vitro and the Thermo Fisher freezer programs operating in School of Veterinary Medicine. And the protein lab, we are featured by protein preparations, peptide, and antibody productions. That will include construction engineering of expression vector, expression and the purification of proteins, synthesis of peptide, and the preparation of poly or mono antibodies. Detailed feature of protein lab will be introduced later on by Dr. Gianluuca. And the protein chemistry and cell biology together, we have some facilities for protein ligand interaction using surface plasmon refractometry and the protein and the small molecule mass specs, flow cytometry, immunostaining, and the fluorescence microbiology, microscopy, metabolic profiling, metabolomics, proteomics, and we also provide biosafety laboratory experiments. And we have also facilities for providing experimental mouse. And the structural biology can do large-scale protein productions and the purifications. That is, we talk about milligram range of protein with 90% or more of purity. And the protein crystallization and screening and optimization attempt, and the data collection and the structure determination from the synchrotron data collection, APS, and the ChemDis. And we also do a little bit of virtual screening and the small size high throughput screenings, and the lead optimization and the structural activity and the relationship study. This one, we can do it, but there is not much need at this point. And protein and the small molecule interaction using NMR study, using LSU NMR facilities. The new development is, I found that there is some need of high resolution cryo-EM studies from large protein complex or membrane proteins and the multiplying complex or viral proteins. This one I have arranged a cooperation from Baylor Medicine. They have very nice cryo-EM core that is capable for all kinds of this experiment, so you can do. And we are preparing now a training program for virtual screening and the molecular dynamics. It's not fully established, but it will be prepared. And the protein post-translational covalent modification is important topic of biomedical research. For that, we are building up a cooperation with the proteomics core of University of Arkansas for medical science. They have like very well specialized core for that. And here we have some list of ongoing work to the MCBR. So there is DNA sequencing, chip DNA sequencing, microRNA sequencing, and a lot of things going on. And there are a lot of people are helping. So I'm not going to go in detail about what's going on right now. New additions. We have added statistical service units. NIH is increasing emphasis on the scientific rigor. To guarantee this scientific rigor, proper statistical approach and analysis is necessary. So we have strengths with that. And we can get help of scientific readings for our manuscript for publication and the grant proposals. And bioinformatics data and storage. So we can use data mining and raw data storage through CCT. This year we have new some changes. Yes. Okay. Dr. Gianluuca Reggiani has joined us as a new leader of Protein Lab. So he will talk about new features of Protein Lab and his strengths. And the fact is that over 60% of our LBRN project is cancer-related. That means strengthening of translational study is necessary. So that's why we have picked flow cytometry and metabolic profiling as an important part of our technology talk. I have special thanks to Dr. Marilyn A Dietrich. She is expert in here, and Dr. Basel Abuaita using Seahorse-based metabolic studies. Thank you very much. If you have a question. No questions? All right. Yes.
So do LBRN investigative fitness company subsidized prices for employees?
So his question is service fee can be discounted. The fee that is not my part. So I can ask, Grant and Gus, hey, we need discount on this, but I cannot give you definite answer. But I think that will be possible.
I think the discount is not possible in can apply the core of
Yes, yes. CoBox is really, really good, leverage you can use to discount your fees. I'm actually trying to find if CoBox can be used for other states. If there is service from other states, I will let you know if that is possible. Any other question? All right, then. Thank you.
Up next, we got Gianluuca, who's Reggiani, who's managing the protein core facility. He's an expert in phage display and protein engineering. I'm very excited to hear what he has to say.
Hey everyone, it's a pleasure to be here. So I just recently joined LSU, and I'm very excited to share with you what we can do at the protein core, of which I'm a manager. So our main mission is to provide our scientific community with services on protein, other literally proteins, recombinant proteins, but also help you analyzing potentially proteins for whatever your needs are. So we move from recombinant DNA technologies to actually protein characterization, but we provide also as a service, and we expect to do this also as a collaboration in case some of your needs are extremely more demanding than a simple service. And we also expect to provide and educate the scientific community with all the technologies we can offer, as well as particular new trends, and really put to your disposal, to the scientific community disposal, our expertise and knowledge in everything we can do with proteins. So we collaborate a lot with GenLab, and through GenLab we can offer the production and engineering of plasmids with desired tags, for instance, or subcloning into different expression systems, moving from bacteria to mammalian or baculovirus expression systems. Indeed, we heavily rely on sequencing, and the capabilities span from regular Sanger sequencing, just for validation, for instance, of your constructs if you clone genes. But this can be is also heavily used in for next generation sequencing, single cell transcriptomic analysis. And in addition to this, the GenLab offers also the ability of analyzing proteins interaction in a multiplex fashion. They have a machine called BioPlex 2000 that allows to analyze arrays of protein, hundreds of proteins simultaneously, very quickly. When it comes from the protein production point of view, as I previously touched on, we are perfectly suited to express recombinant proteins in a number of different expression methods. We can use bacterial cultures. So for regular expression of proteins, this can be done in small scale as well as large scale, up to the crystallography level amounts. We have a baculovirus expression system that allows us to express protein in insect cells. And this will be particularly helpful when it comes to express proteins that are typically difficult or impossible to express with lower organisms, such as E. coli And we can even express protein in mammalian cells very comfortably. So this gives us a wide range of expression systems that will fit the majority of the needs that potentially scientists can encounter in their scientific endeavors. From a protein purification point of view, we're fully equipped to perform a number of different purification methods, ranging from a classical affinity purification to more sophisticated separation, like in this case, where we use ionic exchange chromatography coupled with size exclusion chromatography, and we can separate species that are extremely similar to a very extremely high quality and purity. And this could be extremely useful depending on the type of application you will need to use your protein on. And the scale range that we can offer moves from small scale, so at the analytical level, up to the preparative level in case you need large quantities. From the point of view of protein characterization, we can offer our expertise in a number of different techniques. This can range from analytical chromatography, either size exclusion or any other chromatography type that you might think of, as well as surface plasmon resonance for determining binding affinities, but as well as interactions in general. At the protein core, there's also an ultracentrifuge, so it's possible to run also velocity and sedimentation experiments to better characterize the oligomeric states of your proteins of interest. What we're currently implementing, it's also offering as a service the possibility of generating antibodies for a fee, of course. So at the moment, we're currently expanding our repertoire of antibodies library and proteins library that we can offer, and potentially engineer binders for desired applications. So we use phage display quite reliably for a number of different targets now. And these allow us not only to obtain quickly, relatively quickly, monoclonal antibodies, for instance, but the information that we generate, which is the sequence of an antibody, will make it a renewable reagent for future years. So it's unlikely to incur in some of the problems that you typically will observe with hybridoma technologies, for instance, where if a hybridoma culture gets contaminated or stop expressing the antibody, you will need to start reproducing the cell line itself, and that will result in a waste of time and money as well. Phage display has, among its many advantages, this one is one of the others, or those ones. Another one is that, for instance, unlike hybridoma technology, we can actually develop antibodies potentially, or any sort of binder for targets that are self, that you typically could not immunize animals with. In addition to this, we can also offer consultancy in how to develop your CRISPR screens. At the protein core at the moment, we have a number of CRISPR libraries. We have a few genome-wide CRISPR libraries, not only for conventional CRISPR, but also for CRISPR interference, which is one of the new trends in the CRISPR field. And we can guide you through the process of how to do this efficiently and what could be the pitfalls you might encounter and how to try to avoid them. So I hope that I convince you to convey to you the message that we can offer a lot of opportunities, and we can feed a lot of the needs that typically are needed in modern science. If you need more information, you can check these two websites, the one from the Protein Core and GeneLab as well, where you can find the detailed list of equipment and techniques that are available to the community. And if you have additional questions, you can even easily reach me at my email address. Thank you. Yeah, sorry.
If you want protein to be produced for 2 hours, make the plasmid and bring it to you, or you help us to make the plasmid.
Okay. So Dr. Joyce asked what we could do to express protein for crystallography purposes, whether we need to clone the plasmid or we can help them. I think we can offer both. If you're already set up in your lab to clone the plasmid yourself with your desired needs, you can do it, and maybe we can just discuss and give you an advice on how to do it more efficiently. There's also the possibility that we can clone the plasmid. It's one of the services offered on GeneLab, if you look at the website. So that's clearly, it's a fee service based, but we can easily do it. Okay. Thank you.
Right, and up next, we have Marilyn Dietrich, who's expert in flow cytometry. She runs our flow cytometry facility here at SVM. She'll be talking to us about principles and applications of flow cytometry. So let's.
Okay, today I just really want to explain to you how a flow cytometer works, and then I'm also going to discuss data analysis. It's going to be a lot of fundamentals as well, because I didn't know what the audience, how much flow cytometry you were exposed to. A lot of people are exposed to fluorescent microscopy, so sort of think of a flow cytometer as basically an automated fluorescent microscope, if you're not as familiar with flow cytometry. First of all, let's start with a definition. Flow cytometry is a technology that allows simultaneous measurement of multiple physical and chemical characteristics of a single cell. Basically, the physical characteristics that we look at are mostly immunophenotyping. We want to look at labeled ligand either on the surface of the cell or perhaps intracellular. Immunophenotyping is predominantly 90% of the flow cytometry applications we perform. We also look at things such as apoptosis, as well as DNA cell cycle. The chemical characteristics are typically kinetic assays. If you want to look at intracellular calcium, intracellular pH, membrane potential, then we can also look at that using flow cytometry. The measurements are made on a per cell basis at routine rates of 200 to 30,000 cells per second in a moving stream. We've got a lot of other instruments in the core facility, and also part of another core facility, the PIP core lab as well. So I'm only going to show you the latest, most current purchases that we've made. As far as analyzers go, we have the Fortessa X20. This pretty much went online predominantly in 2018. And it is a 15-color instrument, and it will look at cell size and internal granularity. It's got 4 lasers. We've got a 405 nanometer. We've got a 488, 635, and a 561 nanometer. So 4 different colors we can do. 4 different lasers. We can do 15 different colors using this particular instrument. Now, this is our latest acquisition at the School of Veterinary Medicine. It is a Melody sorter. We just installed this in December of 2022. This particular instrument not only analyzes cells, but it can also sort cells. You can bring a heterogeneous population, and we can put it through this instrument and actually give you a purified population of cells. The instrument itself will do a 4-way sort, but there are technology out there now that actually will do a 6-way sort. This particular instrument has 3 lasers. It has the blue, it also has a violet and a yellow-green laser, and it can do up to 18 fluorescence emissions and, of course, sort 4 ways. Now, regardless if you're looking at an analyzer or if you're looking at a cell sorter, there are primarily 4 different components of any flow cytometer. First of all, you have to have a laser. That's your light source. That's going to excite the fluorochromes, emit the fluorescence in the form of some type of fluorochrome that you've conjugated to a probe. And then secondly, just as you would have to optically align a fluorescent microscope, you must align a flow cytometer. So it's going to consist of a series of mirrors, prisms, lenses, and filters. It's got to capture the final accepted wavelength for that particular photodetector, so that we can capture just that specific fluorescence emission from that particular fluorochrome. Of course, the fluidics is very important, because that is what makes flow cytometry separate from all other technologies, basically most other technologies, is that we are analyzing cells in a single file. And in order to achieve the cells in a single file, we have to establish flow velocities so that we maintain that single file movement. And lastly, we are going to look at the electronics. Those consist of photomultiplier tubes and photodiodes. They're going to take the photons of light, the fluorescent light, or even scattered laser light, and convert that into a current, subsequently into a voltage, and then that voltage is read as a digitized signal using some type of interface graphic software. Now, regardless if you're using an analyzer or a sorter, we also are going to primarily look at 3 different parameters. First of all, forward angle light scatter. That is cell size. Basically, when the cell is hit by the incident laser beam, specifically the blue light, it's going to scatter light in all directions. That light that is deflected at 3 to 12 degrees is very sensitive to how big the cell is. So we're going to capture that with a photodiode. We will convert that into a size signal. Likewise, the scatter, side scatter, or also known as 90 degrees or right angle scatter. That is scattered blue light that's captured at 90 degrees. That's refracted light, and it's proportional to the amount of granules and vacuoles, and regardless of even the nucleus. You know, if that has a lot of convolution compared to a simple nucleus such as a lymphocyte, then you're going to have increased side scatter signals. Also 90 degrees to the incident laser beam, we're going to measure fluorescence emission. And it just depends on your technology. You could, I mean, there's machines that only look at 3 colors, and there's other machines that can look at 64 colors. So that will depend on the technology. Best is to use both forward angle light scatter and side scatter. So that's size versus internal granularity. Best to use those together because we can actually get more information that way. On the x-axis of this particular dot plot, you have forward scatter, cell size. On the y-axis, we have internal granularity or side scatter. This represents human peripheral whole blood that has been lysed by ammonium chloride lysine buffer. And then from there, we can actually just look at the leukocyte. So on the x-axis, as you follow it, the further to the right you go on the x-axis, the greater the size of the cell. The further up the y-axis, the greater the intracellular granularity or complexity. So we can actually separate that into 3 different populations: lymphocytes, monocytes, neutrophils, based on those characteristics. And from there, we can gate on that subpopulation and determine frequencies of specific subsets of those subpopulations. Now, fluorescent measurement, typically detection of labeled ligand, live dead determination, DNA and RNA. And I'll show you some examples in the data of these. Briefly, with cell sorting, for the most part, a cell sorter is an analyzer first. They all have a flow cell. Within the flow cell, you're going to analyze the populations that have been stained with labeled probes. And then once you make that measurement, then you're going to pass the cell in a fluidic stream through the flow cell into a nozzle orifice. From there, the nozzle is actually vibrated at a defined frequency, and subsequently liquid is ejected into air to form these droplets based on that oscillation. And then the droplet containing a cell is applied either a negative or a positive charge, and it's sorted by passing it through an electric field. This illustration shows you what's going on at a cell sorter. So at the very top, where the laser beam strikes the incident laser beam strikes the cell, you're actually going to have fluorescence and scattered light in all directions. And then the cell is going to go through a nozzle orifice, and then it's going to actually come through that orifice. And since we applied a frequency to that nozzle, it's oscillating, we're going to have droplets formed. We're going to actually inject a cell of choice into this droplet, and then we're going to give it either a net negative or a net positive charge. And then we're going to pass that through an electric field. These deflector plates are actually going to be charged between 2,000 and about 4,000 volts. They're going to be charged, and so opposites attract. And so you're basically going to collect the cells in different collection vessels. And these typically have a little bit of some buffer at the bottom in order to maintain viability of the cells while we are sorting them. This represents a 2-way sort, but as I said before, our technology allows us to do 4-way sorting. So now we're going to jump into the application so you can actually see what the data looks like. We'll discuss immunophenotyping, DNA cell cycle, apoptosis, and cell function. Just very briefly on preparing cells for flow cytometry. You absolutely have to get your cells into a single cell suspension. Or you'll plug the machine first of all, and then secondly, it defeats your purpose of using a flow cytometer if you don't have single cells. And the rest of this just absolutely represents if you had to perform a indirect immunofluorescence technique versus a fluorescence technique. Since we're over here, we're at a veterinary school. Sometimes we're limited in the probes that we can use for flow cytometry. So sometimes we have to incorporate indirect immunofluorescence, where you add the antibody first, followed by a secondary antibody that's conjugated to a dye. But for the most part, with a lot of the species that we work with in the animal models, we're capable of just using directly conjugated. And then from there, you can either do analysis on the analyzers or cell sorting. This just represents a panel selection. Whenever you want to perform flow cytometry, let's say you're working with tumor cells and you want to find the immune cells within those tumor cell samples, then you're going to have to provide the appropriate panel of antibodies if you're trying to target a lymphocyte subset or you're trying to target macrophage subsets. So you're going to have to look at a list of lymphocyte antibodies and myeloid antibodies and determine your panel of choice. Now, briefly, I will tell you this part is very important because the dyes that you use they can overlap into each other's photodetector. So that takes a little bit of art to figure out exactly which antibody conjugated to which fluorochrome are your method of choice. So take time in designing your panels, and I can help you with that as well. So let's jump into the data. The first one represents immunophenotyping of feline lymphocytes with CD4 monoclonal antibody as well as CD8. So on the x-axis, we have CD4 staining, and then on the y-axis, we have CD8 staining. This cat was presented with a lot of opportunistic infections, was determined to be FIV positive. Therefore, it did look like it was in fully blown AIDS. So on the x-axis, on the x-axis, we have CD4 expression. Likewise, the further to the right you go on the x-axis, the stronger the CD4 expression. The further you go up on the y-axis, the stronger the CD8 PE expression. So these cells up here in the upper left quadrant represent our CD8 positive cells at 55%. Our CD4 cells in the lower right quadrant, 16%. And therefore, if we looked at the ratio of CD4 to CD8 cells, we've got about a 0.29 ratio representing definitely fully blown AIDS in this particular cat. Now, the next example I want to show you is a DNA cell cycle histogram. We have quite a few people that do DNA cell cycle. It seems to come in cycles itself. We'll do it for a while and then we'll go years without anybody doing it. And then all of a sudden, there's another round of people very interested in this. For the most part, these cells, this is a canine melanoma. This represents a tumor that was dissociated into single cells. And we have to actually permeabilize and fix with 70% methanol so that we can get the probe, which is, the probe, which is rubidium iodide. We have to get that in. That only stains dead cells. It won't go into live cells. It's excluded by the membrane. So we have to permeabilize and fix with methanol, and then we can get the PI in. PI intercalates with double-stranded DNA and RNA. So we add some RNase enzymes in order to digest the RNA, the double-stranded RNA. So we're free to just label the double-stranded DNA. So if you look at this plot, along the x-axis, we have DNA content. The further to the right, the stronger the rubidium iodide staining, and therefore more DNA. On the y-axis, just number of cells. This very first peak represents our G0, G1 component of the cell cycle. It's about 60%. So here's your resting cells and cells that are getting ready to go to prepare for DNA replication. And then the secondary peak over here, the smaller one, represents our G2M. These are cells that have already duplicated. They're ready to, they're undergone mitosis and they're getting ready to divide. That's about 27%. So anything between those two peaks is our S phase, DNA synthesis, and that's about 13%. So we can, I usually do this with specialized software that does DNA modeling. I don't typically use it with the regular software on the instrument, but we can actually get very good results with a very standardized technique. As far as S phase, anything above 6% is highly aggressive. So this melanoma represents a very aggressive tumor. Apoptosis, there's lots of ways to do apoptosis using flow cytometry. You can fix some procedures such as tuttle required fixation. I like this particular application the best, this because it's using live cells. Basically this is a Nexin-5 staining. A Nexin-5 is a phosphoprotein that has an affinity for phosphatidylserine. Phosphatidylserine is normally located on the inner leaf of the cell membrane. However, this phospholipid actually gets expressed to the outer portion of the membrane once programmed cell death is initiated. So we're going to look at a Nexin-5, which is going to tell us if phosphatidylserine is exposed on the outer portion of the leaflet. And then we're also going to look at propydium iodide on the y-axis. Now we're using it with live cells. So if we see propydium iodide staining, that means our cells are dead and we have a leaking membrane. So if we look at a dual parameter plot of a Nexin-5 versus propydium iodide, the lower left quadrant, that's going to represent totally negative cells for a Nexin-5 or PI. There's your viable cells. Those are live cells, 33%. And then if we look at the lower left, the lower right quadrant, those are cells that only will stain with the Nexin-5. That is about 35%. So those are our apoptotic cells. If cells are double positive, meaning they are expressing the Nexin-5 probe attaching to the phosphatidylserine, and also they're picking up propydium iodide in their membranes, they're necrotic. And that is about 31% of the population. Now, the next slide, I just want to show you a little bit about cell function, some cell functional assays. We do a lot of cytokine detection, intracellular cytokine. In this particular example, we have a murine footpad that has been inoculated with Mycobacterium leprae. And typically, they have to do this in order to get enough M. leprae to actually do one experiment. So it was inoculated for about 6 months. And then we went into the footpad and we looked at what cells were there in that particular area with the M. leprae colonization. So in this first plot, we're just looking at internal granularity or internal complexity versus CD4 expression. And the majority of these cells are macrophages. They are not picking up any of the CD4 cells, and they're actually quite granular. Internal granularity. So those are your macrophages. These are probably just CD4 negative lymphocytes and other cells. And then here's our CD4 positive cells. We're going to set a gate around the CD4 positive cells, and we want to look at CD44 expression versus interferon gamma. It was pretty much a pretty big panel that we worked with, but I just want to show you the data that look good. CD4 expression is normally found on lymphocytes, but low expression. If the cells are activated, stimulated, then we're going to have upregulation of CD4. And as you can see, very bright CD4 staining. So the upper left quadrant represents just CD4 expression, very intense CD4 expression, and that's 58%. Now the upper right quadrant is going to represent dual positive cells. Not only do we have upregulation of CD44, but they're also expressing interferon gamma. So that's about 38% of the cells. So basically, there's a lot of stimulation activation going on in these particular cells. And some of the other cytokines we use, we were just trying to find out if it was pro-inflammatory or anti-inflammatory response. And as a result, definitely a pro-inflammatory response. Now, this next particular slide is going to illustrate to you how we perform more more immunophenotyping in a more complex environment. This represents multicolor staining. We're not working with two colors. We're working with definitely more than two colors here. This represents adipose tissue. We're trying to find, we're trying to characterize M1 and M2 macrophages in adipose tissue. So at the beginning, we're going to start looking at all the cells. And then we're going to start with just looking at size, the area of the size measurement versus the height of the signal for the size measurement. And this represents single cells in this rectangular window. And then from there, we're going to look at a viability dye. We're going to take the cells that are negative for DAPI, the live cells. We're going to look at a scatter plot of size versus internal granularity, eliminate debris. We're going to come over here and we're going to gate on CD45, a pan-leukocyte marker, which is only going to pick up our immune cells. We can throw away all the epithelial cells that are found in adipose tissue. And then we're going to look at CD11B versus F480. That will label any myeloid cell just about. But we take the very brightest population and we look at CD301 versus CD11C. We've got M2 macrophages at 77.4% and M1 macrophages at 13.1%. So we were able to characterize that for the macrophage population. Okay, I'm going to cut it off here. Advantages, precision, very sensitive, not as sensitive as ELISA and some other applications, but definitely more sensitive than fluorescent microscopy. No photobleaching or quenching, volume. You can typically, I tell people, very good concentrations, maybe 40 to 60 cells per hour. Samples per hour. And then single cell analysis, very powerful. You don't get an arithmetic mean with flow cytometry. You actually get single cell analysis. Sampling statistics include other things besides percent positives, and the ability to sort in separate tubes is a very powerful application. Do I have any questions?
After you do DNA based profiling, can you separate the cells in different cell cytoscape?
You could. Yes, you can sort them. Yes. Normally we do sort on fluorescence, but I have actually sorted even just based on size and internal granularity, depending on the tissue that we're working with. Yes. Pretty much flow will do anything for you as long as the size is adequate and you can find a probe that's conjugated to a dye. You can run it through flow cytometry. Okay, thank you.
Up next, we have Basel Abuaita, who's a professor in the Department of PBS. He's going to talk to us about the applications of Seahorse, which is used for
Thank you, Brett, and thanks to Dr. Lee for giving me the opportunity to tell you a little bit about, this kind of technology that the Seahorse, or how can you actually engage metabolisms in your research? How can you assess the role of metabolisms in your research? And currently, we have two analyzers that are hosted in pulmonary immunopathology core. We have an 8-well analyzer as well as 24 was a 96-well analyzer. The 8-well analyzer is good for if you have a precious sample, so you don't have a lot of replicates, or if you don't have many different conditions. The 96-well analyzer will give you an advantage of using many different conditions as well as several replicates. And if you are interested in actually performing some of these research, but you don't know where to start, then just please feel free to contact me or contact Dr. Tiro as well, as he maintained those instruments. All right, so for historically, metabolisms and immunology have been distinct fields, but nowadays we know it's different in a way that many of the metabolisms can directly impact in immunity, as well as immunity can impact in metabolisms. Not only metabolisms can impact immunity by just basically providing energy, but several of these metabolites can actually drive a specific inflammatory response and can control the outcome of these disease. At the same time, many inflammatory signals like interferons and as well as cytokines, chemokines can actually drive metabolisms to control the outcome of this disease. And one concept that's actually is emerging is that can we modulate metabolisms in a way that we can enhance or enhance treatment or for treatment of inflammatory disease. And in order to do that, we have to actually find a way to analyze metabolic flux in our experiments. And that's what the Seahorse analyzer will allow us to do. And so the Seahorse analyzer can do two things. One is that can measure two things. It can measure oxygen consumption and extracellular acidification. It's that simple. And but then we can use those measurements to calculate mitochondrial respiration. And that's basically because we actually measuring the oxygen consumption as well as glycolysis based on extracellular acidifications. Once we set up, if we want to set up an experiment, there is two steps that involve, two major steps that's involved in that process. The first one is basically set up your experiment. And that's the same things that you set it up like any other experiment. So you, for example, can isolate cells from a mouse and see them in a well. You infect one and you leave one uninfected, and you sort of compare the metabolic rates between them. And that the time-wise, it depends on your basically your experiment. The second step is basically performing the assay, which basically you can just throw it in the machines, and now we can isolate or measure oxygen consumption as well as crystal acidification. And there is a lot of different assays that you can run. One assay is, for example, mitochondrial stress response or stress test. You can measure in specifically the capacity of these cells, measuring mitochondrial metabolisms, as well as the glycolysis, for example, the capacity of the cells that's doing glycolysis and so forth. And so here I'm showing the steps and the timelines of these assays. So initially, an experiment setup, and that's actually vary, and the time is varying here. So it depends on your experiment setup. So you can actually isolate either a wild type cells from wild type and knockout mice, put them on the plate, whether the mice has been infected or non-infected or stimulated, or you could isolate tumor cells, for example, from a mouse and see them on here, and then once that is done, then you can move on to the actual assay, and that usually take about, preparations of the assay usually take about an hour, but then after that, it depends on the assay. It can go range from 1 to 2 hours. And then after that, you could just simply analyze the data and then have a readout. And so what is actually the machine is doing is that it's basically, it's measuring, as I said, oxygen consumption and extracellular acidification. And so it takes a 3 measurement first and then inject drugs, and it depends on the assay that you are running, and then take an additional measurement and so forth. So for example, these mitochondrial stress response, stress test, what it's doing is taking oxygen consumptions 3 times points, injecting oligomycins to inhibit ATP synthesis, and then that dropped the oxygen consumption. And then injecting another drug called FCCB, which enhance oxygen consumption. And then you could detect the rate of oxygen consumption in, for example, mock cells versus infected cells or stimulated cells or drug inhibiting cells and so forth. So now how can this is applicable? One of the area that's actually the Seahorse has been really excessively used is studying the macrophages function. So we know that metabolic programming is critical for macrophages function. Macrophages does exist in at least 2 different states. One is inflammatory state, what we can refer to it as an M1 state, and the other one is M2 state or tissue repair state or anti-inflammatory state. And we know that when we stimulate those macrophages with inflammatory cytokines or infections, we drive them to an M1 state, and those depends on more on glycolysis, less depends on oxidative phosphorylation, and those kind of things is important for driving these anti-inflammatory states. On the other hand, is M2 is anti-inflammatory state. They actually tend to depend more on oxidative phosphorylation rather than glycolysis. So an emerging field is like, can we edit macrophages polarization or state in a way that to enhance treatments for inflammatory disease? And well, it was cut off.
Stream, yeah, it's static.
Oh, sorry. Okay, so for some reason it was moved on. Anyhow, so I'll explain without, you have to trust me on this one, but here is the reference for this. So what this group did is basically they stimulate macrophages for M1 stimuli, and then they can see that that decrease oxygen consumption. And then you can stimulate it with M2, like IL-4, then that drive it to an M2 state. And the question is, can we reverse it? Can we make the M1 states go back to M2 state? And if you do that, if you stimulate it with the M1 stimuli and then M2 stimuli, it does not. But if you inhibit, for example, INOS, it does do that. So suggesting and doing this by basically using the Seahorse technology. So basically now we can find a way that we can tweak an M1 state macrophages by inhibitions of INOS production into an M2 state. All right. So another one is important. Assay is monitoring rate ATP productions, and this is probably one of the simplest tests that you could actually use, and from that test you could actually calculate the amount of ATP that is produced by the mitochondria as well as by glycolysis. And actually we have used those kind of tests in our research. One of the questions that I asked when I came here is that, can LBS that's driven from a different strains of bacteria stimulate the same state of metabolisms? And so and the reason we went that route, because some of the Salmonella typhimurium LBS are different. And so Salmonella typhimurium exists in many different serovars. But the main difference between these serovars, which they call these different disease, the main difference between these serovars is the LPS. And we know that when we infect macrophages with these different serovars, one serovars produce robust inflammatory response, but then the other ones, they don't. So, and since the LPS is different, so we asked, do they trigger different metabolic outcomes? And so here I'm showing those Seahorse data. What we can see is that upon stimulations with the LPS, either from Salmonella typhimurium or Salmonella enteriditis, we can actually increase oxygen consumption as well as extracellular acidification. And we can quantify how much is come from glycolytic as well as mitochondrial ATP. And what we can actually see from this graph is that both LPS, that's either from typhimurium or enteriditis, tends to actually enhance ATP production, and it seems to be mediated mainly through glycolysis. So we can get those kind of results, basically. So another area that's actually been actively using the Seahorse technology to define T cell function, basically, especially during cancer. And that's because we know that T cell differentiations and activations has a different metabolic state. So for example, naive T cells has an OXPHOX, more OXPHOX than glycolysis. Effector has more glycolysis on OXPHOX, and then the memory has more OXPHOX than glycolysis. And okay, so for some reason these are moved probably from anyhow. So basically, this group shows that basically you could isolate CD8 T cells from naive mice, grow it activate them, grow them under a complete medium or a medium that's lack glutamine, and then take those CDT cells, and then you shows that those have different oxidative consumption as well as different metabolic state, basically. And so they take those things and put them in the mice and shows that those T cells that's been cultured without glutamine tends to actually restrict tumor size and this sort of things showing. Unfortunately, it somehow has got moved. I don't know why this is the case. So anyhow, so if you're actually interested to see that, that's the beautiful. I thought it was beautiful paper is done by Nappy. Anyhow, all right. So lastly, I wanted to say that you could actually allow the drugs be injected by the machine. So and here is an example showing that. So basically, you could isolate T cells, put them on a plate, and then allow the machine to inject, for example, an antibody that activate T cells and measure their metabolic changes. And so you could actually, see as a basically proliferation, similar to the proliferation assay of a T cell. Anyway. All right. So with that, I'll have the like the thankful for some sort of fundings, since either from the lung COBRI as well as the department funding, and happy to take some questions.